Sorry. So, without further introductions, I'm going to start sharing the first presentation, which is about diving. Well, breathing and diving. The class today, as I said, will have two components. One of them is diving and the other is echolocation, and these two are very connected, and I'm going to briefly say how they are connected. So let me just put this in presentation mode. I think you are seeing the presentation now, right? Okay, good. Okay, so I'm going to jump a little bit because this was a longer presentation. I'm going to just go to the short points, but I think this small video is interesting. Let me know if you can hear the sound.
I don't have the sound personally.
Can you hear the sound, Hugh?
No, I don't.
Oh. Oh well. Well, anyway, I can probably do some because I'm hearing the sound. It's never mind. So the thing is, they're talking about the champions in diving. So let me just give me a second. Let me take up the sound. So they're talking about the champions in diving in the marine mammal world, and of course, this is an image of a sperm whale diving. Sperm whales are the deepest diving animal as far as we know. But of course, in the second one, at least on this video, we'll see that this is not so simple. But in this video, they present elephant seals as the second biggest diving, deepest diving animal. And both sperm whales and elephant seals can stay underwater for about 2 hours. 2 hours without breathing. And then the Weddell seal is also a champion. Weddell seals do not dive so deep. They dive around 700 or 800, up down to 700 meters, and they can stay more than 1 hour underwater. They dive for half a day, and then they have to rest on the ice for the rest of the day. So they dive for about 12 hours each day, and then rest on the ice for another 12 hours and repeat the cycle. So obviously then, not all marine mammals, we're going to talk about this, not all marine mammals are big divers, but diving has a connection with echolocation. And this paper here is very interesting. This is, well, this is a paper from 2007, which I just found recently, always looking for evolutionary explanations of things. And in this, these authors present a case for the evolution of echolocation. And according to their hypothesis, which they test and it sounds solid, echolocation evolved as a way to find prey in the night. Okay, for those of you who have had some oceanography, biological oceanography in the past, or have read something about this, there's a very important pattern in the oceans where you have well, you have the primary production at the surface, so you have all the algae and the so you have all the phytoplankton and the zooplankton at the surface, but also you have all the major predators, tuna fish and swordfish, also at the top of the ocean. So you have this massive biomass of fish and cephalopods that are in midwater, so they are in the euphotic zone, so a zone of obscurity, in a crepuscular zone at 200, 300 meters depth during the night, and they go up to the during the day, and they go up to the surface during the night to feed. And so the idea is that the echolocation evolved in cetaceans as a way to locate animals, locate prey during the night. Okay, so there was presumably a selective pressure for animals that could find prey during the night, because this is when all these animals come to the surface to feed, and these authors claim that this prey was basically cephalopods. So so the idea is that echolocation evolved first as a way to find prey in the night, and then the diving capacity evolved after this, once the animals had developed a way of finding prey in the night. So they were not, they could track prey as they go down, as they dip, as they dive during the day, and they would continue eating during the night, in depth. So it's an interesting theory. Echolocation evolved first as an adaptation to feeding during the night, and then deep diving evolved as animals then went down and down and down, looking for that same prey, and interestingly, cephalopods. And today, as you can see here on this slide, the deepest diving animals, deepest diving marine mammals, I mean, are cephalopod eating. So they eat squid, basically. And you can see here the beaked whales, and so the first two slides are for beaked whales, and the second slide is for the sperm whale. And you can see this is actual diving patterns recording during 24 hours, okay? And you see sperm whales regularly diving to 1,000 meters or more for periods, for long periods, about 1 hour something, and then coming back to the surface. And then you have shorter dives, until the animals make deep dives again. So and as I was saying, they are all feeding on cephalopods. And there is also no clear night and day patterns. For instance, in this case, in the cephalopods, sorry, in the sperm whales, they dove during the whole 24-hour cycle. Of course, not all echolocating animals are deep divers, and so, for instance, orcas are not only not deep diving, but they also do not make long dives. And so this fits in with the hypothesis that echolocation evolved first, and only then in some groups, those groups that continue to be specialized in cephalopods, was the capacity to make long and deep dives evolved. And also in pinnipeds, we see the same pattern. We have the elephant seals here, diving for long periods to deep waters, whereas you have other, in this case, other otarids, which dive to very, very shallow waters and for very brief periods of time. Okay, so that was the first point I wanted to make: the evolution of echolocation first, and then deep diving. And of course, this diagram here tries to call the attention to this fact, to these adaptations of marine mammals to diving. What we see here is that there is a relationship between body mass and the capacity to hold their breath. Okay, and you have a relationship there. You see other mammals, so you have that first line there that shows that there is a correspondence. So bigger animals can hold their breath longer than smaller animals, and this happens with all mammals, right? Note that the scales are logarithmic. So, for instance, sirenians, I mean, are really in the middle here. So they're not different from other mammals in terms of holding their breath. So they're right in the middle. And animals like sea otters or dolphins or even the big whales are just relatively better than other animals at diving and breath holding capacities. So they have adaptations, but they're not extreme. The extreme adaptations you will see in sperm whales and in beak whales. But the really big adaptations are in seals, particularly the elephant seal, because for their size, they're also on the 2 hour, 1 and 2 hour breath holding capacity. And this requires big adaptations to animals which are 3 orders of magnitude smaller than sperm whales. What I mean is, it's much more difficult for elephant seals, and elephant seals have to have had a larger, more comprehensive evolutionary adaptation to be able to dive for so long as they do. Okay, so now what are the challenges? And I'm going to cover this relatively quick, but just to point out that diving for long periods is problematic for 3 reasons, basically. The first one is the accumulation of subproducts from cellular respiration, right? So when in our case, if we try to hold our breath and not to hold our breath, what happens is that our oxygen reserves get spent, and if we continue holding our breath, then what we have is an accumulation of lactic acid. So many of our cells just switch into anaerobic respiration, and then you have lactic acid accumulation, and this can lead to exhaustion, and eventually to well, we need to breathe again. So that's one thing, the challenge, and I'm going to. So what marine mammals evolved was a capacity to continue breathing aerobically, although they are not ventilating. So that's the first point. The second point is diving and depth has an effect on all spaces with air. So water is incompressible, liquids are incompressible, but gases are affected by pressure. So when animals dive, all the spaces in their body with air, starting with the lungs, are affected by this, and the volume decreases. And so you need adaptations for this. And the last thing is, under pressure also, you have changes in the gas content in the blood, and this may lead to problems of oxygen toxicity, problems with nitrogen toxicity, and also decompression sickness. So let's I guess some of you do not dive, do you? Do you scuba dive?
Not regularly.
Sorry?
I did, but not. Yes, no. I do.
Well,
I do not.
So you probably, if you did some training in scuba diving, then you've probably heard some of these issues, right?
Yes. Yes. Yes.
And I imagine other people here will have the same experience. So let's go briefly into each one of them. The first one has to do with the aerobic metabolism. Okay, so previously there was this idea that, well, some years ago, there was this idea that the animals that were deep diving would have elevated concentrations of lactic acid in the blood. Okay, so the same thing by analogy with what happens to us. I mean, if we go running, depending on how trained you are, you at some point you will start accumulating lactic acid. So your muscles are spending oxygen faster than you can replenish it from the air you breathe, and so muscles the muscle cells will start shifting into an anaerobic metabolism, producing lactic acid. And this lactic acid accumulates in the blood, and you'll start feeling tired as you're running, and then eventually you have to stop. And this can even lead to cramps. Is that the term when you have pain in the muscles? Cramps. So this model was assumed to happen also with marine mammals. What we have learned since then is that actually marine mammals do not go into anaerobiotic metabolism in their dives normally. So in this case, this is the I'm not sure if this is the Weddell seal that we have here on the screen. I don't remember anymore. But the point is that they've measured the level of lactate in the muscles of these seals after a dive. So they would dive and they will come up to the surface and they would in some way record the lactate level in their blood. And what they saw was that if the dives were up to 20 or 30 centimeters, there was no change in the lactate levels. Okay? It was only for dives that were longer than 30 minutes that the levels of lactate would rise above the normal resting state. What this means is that when this species dives to 20 minutes, she has no problem with lack of oxygen. She continues her normal oxygenated metabolism. Okay, only if the dive is longer will they start shifting into anaerobiosis and then producing lactate. Okay, so this is called the aerobic dive limit, and presumably every species has one. Okay, our aerobic dive limit would be 30 seconds or something like this, or 1 minute, depending on the people, but it's ridiculous. So it's a bit awesome for us to think that an animal can not ventilate for 20 minutes and still do not lack oxygen. And of course, sperm whales, their an aerobic diving limit would be something like 1 hour. Okay, so they are not, when they come up to the surface, they are not like when we come up to the surface, grasping for breath. They're just normally. For them, 1 hour underwater is just holding our breath for 10 seconds like we do. Okay, so that's one point. So where do they have this? They must have huge reserves of oxygen, right? Where do they have them? Well, they have them in two. This slide would need to be inverted, so we needed to start from the bottom. So there's basically three, and let me jump to here. Okay, so there's basically three reservoirs of oxygen. One is lungs. Okay, so in our case, lungs are here represented by the gray area with the L. And so, for instance, in our case, most of our oxygen when we dive, or when we hold our breath, most of our oxygen is kept in the lungs. So when we want to dive longer, we just really fill our lungs to be able to take the oxygen with us down. And the rest of it is in our blood. Okay, there's another reserve of oxygen which is in the muscles. Okay, like blood, like the hemoglobin in the blood, which is the pigment that retains oxygen, we also have myoglobin, which is a very similar protein, but is stored in the muscles. Okay, so and this is what gives the muscles their red color. So we have relatively modest reservoirs of myoglobin so of oxygen storing pigments in the muscles. If you look at the bottlenose dolphin, for instance, just the other side of the humans, you will see that the importance of lungs as storage of oxygen is reduced. And if you go to the sperm whales or the Weddell seal, you will see that lungs are residual. So the amount of oxygen in the lungs is negligible. It has no importance. In fact, seals, pinnipeds in general, exhale before they dive. It's an interesting situation. Cetaceans inhale, so they dive with their full lungs, but actually pinnipeds exhale, they take the air out of the lungs before they dive. Because in either case, it's not relevant. It's not where they have the big storage of oxygen. The big storage of oxygen in seals is in the blood. In cetaceans, and particularly in big diving cetaceans, the largest storage of oxygen is in the muscles. And so I'm going to move up the scale. So this is a slice of dolphin meat for sale in a Japanese supermarket. And you see the, of course, well, the point is I chose this slide to show you the color of the muscle. I don't know if you have this idea. Cetacean muscle is very deep red, almost black. Okay? The reason for this is the huge concentration of myoglobin. And because they store most of their oxygen in the muscle. So when the animal dives, the muscles can continue working using the oxygen that is stored within themselves. They do not need so much the oxygen in circulation from the body. And in fact, the body circulation is cut to the muscles and shunted to the brain. Of course, seals, on the other hand, which have massive storage of pigments in the blood, they have very important adaptations in terms of the amount of blood in circulation. Okay, so they have a system of sinus. So they have reservoirs that fill with blood when the seal is not diving. They have what they call rete mirabili. So they have areas in the body where there's huge concentration of blood vessels, and these are also storage of blood. Okay, that then when the animal dives, this blood enters into circulation and takes the oxygen to wherever it's needed. Okay, so it's two convergent paths, but cetaceans adapted to myoglobin maximization, where pinnipeds opted for hemoglobin maximization. But the result is the same. They have capacities for deep diving. Now, the other thing is the second point is what happens to air, right? So, Hugh, if you had some, when you're diving, either free diving or scuba diving, when you dive, what is something that is essential that you do when you start diving?
We have to eject air with our nose.
Yes. Why?
In order to make that the water won't go through our nose, and yeah, because we don't want water in our body.
Don't you have to, when you dive, don't you have to compensate? Doesn't something happen to your ears?
Ah, yes, yes. Because of the pressure, we can have something happening in our ears, and it's painful.
Yes.
so we have to uh, I don't know how to explain in English, but to I don't know how to say, but we have
You have to,
to,
yes, you have to compensate. So what you do is you press your nose and you
yes,
blow and you feel something in your ears and suddenly the pressure is relieved. Okay.
yes, that's
So
it.
do you have any idea of what the mechanisms that are involved here?
The air, I think, I don't know, but it's, yeah, what I think. The air which is behind the, in the ear,
Yes.
um, uh, has to escape, or no, because of the pressure, it takes less space. So we
Yes,
have to equilibrate,
that's it.
and yeah, so air comes up to the ear, and
Yes, the thing is, uh, 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 we have air inside our ears, and when we dive, all the compartments with air shrink in size. Okay, so that's the first point. Our lungs shrink. Okay, if we dive to say 5 meters, our lungs are a quarter of their size when we are at the surface. Okay, if we dive to 10 meters, our lungs shrink to half of the size that they have at the surface. And the same thing happens with our ears. So there's a small pockets of air, very small, but ears are very sensitive. And so when we start diving, these pockets of air are compressed and it hurts. It hurts like hell. It's impossible to dive if we do not compensate. Fortunately, we have a link between our nose and our ear. And what we do is that we blow air through those little channels inside the ear, and then the pressure equilibrates and it stops hurting. So we cetaceans and marine mammals do not have hands, okay, so they cannot do this. So what happens is their connection with the with well, two things happen. One of them in cetaceans is that they do not have air at all inside the middle ear, and so this is not a problem. Okay, I don't know, sincerely, I don't know what the situation is with pinnipeds, but I imagine I think otarids have little ears, so I don't know what their adaptation for this is, but it may involve a larger connection with the mouth and nose, making this equilibrium automatic. Anyway, if you're diving to 200 meters or 300 meters, your lungs collapse completely. Okay, so one adaptation. So that's another thing. Um, all marine mammals that dive deep have adaptations to be able to cope with the fact that the lungs collapse completely. Okay, they're completely flat. All the small airways and como é que se diz, alvéolos pulmonares. Those little sacs, I don't, skipping the name in English, those little sacs at the end of the respiratory tree, they collapse, everything collapses. So you need to have two mechanisms that allow these surfaces to open again when the animal comes to the surface. And we need to have muscles and cartilage that will be able to collapse and then resume the normal structure. So the big divers have adaptations in their lungs to withstand these complete shutdown of the respiratory surfaces. And the last thing has to do with gas, the effects of gas, different gases in the bloodstream. So now this is a bit complex, and I think I have too many slides here. But let me see if I can explain this briefly. Of course, people who have take a scuba diving course will know about this, will have learned that there is a first thing is as we dive, as the air gets compressed, the air in the lungs gets compressed, and so this means that the partial pressure of the different gases increases. So at 10 meters the pressure is twice that the atmospheric pressure, and this means that um, the amount of a gas, say nitrogen, per unit of volume is the double. So this means that the partial pressure of nitrogen is multiplied by 2, and then at 20 meters, at 3 atmospheres, the pressure of nitrogen is multiplied by 3 and by 4 and by 5 as we dive, or as the animal dive. And so that's the first point. The second point is whereas oxygen, we keep consuming oxygen, nitrogen is an inert gas. So no mammal uses nitrogen for anything. It's just there. It comes in, it goes out, no problem. And so there is no mechanisms for controlling the amount of nitrogen in the blood. It would be impossible to do this, but anyway, there aren't. So when animals dive to 2,000 meters, they are exposed to huge partial pressures of nitrogen, and this means that this nitrogen could potentially go into the bloodstream. Okay, that's not a problem in itself. That is a problem. Let me just move a little bit further here. That is a problem not when you go down, that is a problem when you go up. And in this slide, what you see is the classical mechanism of decompression sickness. Say you are a diver and you are breathing compressed air at 30 meters for half an hour. Okay. When you come up to the surface, so this means that your blood is accumulating nitrogen at 3 times the concentration you have at the surface. Okay. But for the moment, let's see, at 30 meters, that's not a problem in itself. It is a problem when you come up to the surface. Because if you come to the surface very quickly, then it means you reach the surface with 3 times the amount of nitrogen that are normal. And this means that the nitrogen will then form bubbles like when you open a bottle of beer or a bottle of any refrigerated drink. When you release the pressure, then you have bubbles forming because of all the compressed gas that was inside the bottle. So this is a very dangerous situation. You do not want bubbles of gas inside the bloodstream. They will clog your capillaries. They will cause shutdown of blood to the brain or to muscles, terrible pain, and so it's a very serious situation. Divers solve this problem by ascending slowly and giving time for the nitrogen that was dissolved in the blood to go back into circulation and be eliminated by breathing as the diver comes up. So the solution to this problem is a simple one. You go up slowly, sometimes even stopping at certain depth to allow time for all this nitrogen that was dissolved to come out. Okay. Now again, what does this have to do with let me see, no, never mind. What does this have to do with cetaceans or pinnipeds, deep diving marine mammals? Initially, it was thought that because the lungs collapse when the animals dive, like you have here, this is an image of the best image I could find, and it's not a big one, of a dolphin that is supposed to show behind the fin, behind the flipper, you see a cavity, and this cavity is caused by the collapse of the um, ribs. The rib cage just collapses. That's another adaptation, by the way, of cetaceans. They have articulate ribs and they just fold. So now, because of this, it was assumed that deep diving cetaceans or pinnipeds would not have decompression problems because their lungs collapse, right? So there is no exchange of gas in diving underwater, and so there is no problem of increasing the amount of nitrogen in the blood. I'm sorry if this is too fast for some of you, because these are complicated concepts, but I hope that you can read something about this, and if you have any questions, you can always ask me by the normal channels. So initially it was thought that as I said, deep diving animals could not have decompression sickness. Okay. Now, this slide here shows, this is from a paper from a 2006 or 8 paper showing mass strandings of beaked whales. Now, beaked whales are very shy animals. They are completely oceanic. They are very hard to see because they are always in very deep waters away from people. And most of the time, as I said, they are shy. Several of the beaked whale species are only known from stranded animals, animals that died and washed up on a beach, and then somebody was knowledgeable enough to say, well, this is a different species. So they are very poorly known because of this. And so when you have a mass stranding of beaked whales, that is highly unusual. Okay, and so and so the research done on these mass strandings of beaked whales as shown, and this is also from the paper, and it shows the locations of mass strandings of beaked whales. And the interesting thing is, you can notice there is that you have a concentration of beaked whale strandings in the Canary Islands, right? So you had a mass stranding in 1985 of 13 animals, and then you had another one in 1986 and 1988 and 1989 of 20 animals, and then again in 2002 of 14 animals. So suddenly there was this massive strandings of animals, and the researchers started wondering why on earth were these animals dying. Okay, and when they brought the animals back to the labs and did the necropsies, what they found was massive internal hemorrhages caused obviously by decompression problems. So these creatures were dying, their ears were blowing, their lungs were shattered, their liver, all internal organs, or the vital organs, were just literally burst with bubbles of air, of gas. And so and they realized that that's number one. Number two is that they realized that this was correlated with naval exercises. So every time there was this massive stranding of beaked whales, it coincided day by day with exercises done by the Navy. Okay, and the NATO, so the NATO Navy from the Western countries, United States and Europe, they were conducting on a regular basis exercises off the Canary Islands. And every time they had one of these exercises, there was a mass stranding of beaked whales. Of course, the two things had to be connected. And the link to those is, this is a complicated model, but I will finish with this thing. What this showed was that beaked whales being deep diving cetaceans and spending so much time in deep water are not immune to decompression sickness. And the model we have now is something that is on this last slide. And the idea is, yes, when animals are in deep water, their lungs are collapsed and there's no intake of nitrogen. But until they get there, there is. So as the animal is diving, the nitrogen is being dissolved in the blood, so that, of course, when the lungs collapse completely, this stops. But then the animal moves up to the surface slowly, but in any case, fast enough to reach the surface in a saturated state. So the idea now is that sperm whales, and particularly beaked whales, are in a permanent state of hypersaturation in nitrogen. Okay, so and they deal with this physiologically, so it doesn't affect them in their normal life, because they're going back again in a few minutes, and they're spending their time at very deep water. But when we have these massive noises, underwater noises made by the Navy exercises, and particularly what they call the active sonars, the animals are frightened and disoriented, and their first reaction is to swim very fast to the surface. And this is what caused decompression. There is also the theory that there is also the theory that there may be other mechanisms involved, but this also shows that the deep diving animals, the marine mammals, are not immune to decompression sickness. Okay, so where are you?
I have one question.
Yes, if you have any questions, yes.
If the bubbles are formed in the blood when they are in the surface, but then they come back down, it solves anything, or no, the problem remains?
Bubbles only formed, if bubbles are formed, that's a big problem. So in a normal situation, bubbles do not form. Okay, so the animals come up, so they have a hypersaturation of nitrogen in their blood, but only up to a level where bubbles do not form. So it's the only problem is if they come up to the surface very quickly, where the difference between the amount of nitrogen in the blood and the outside pressure of nitrogen is too big, and then the bubbles form, and that's the problem.
And then if they go back down the water, it's the same. When the bubbles are formed, you can't solve it.
When the bubbles are formed, what, sorry?
Maybe it's not a good question, but if the bubbles are formed because of going up very quickly, yes, and then you go back to the water, it can re-dissolve in yes, the blood? Okay.
Yes. Yes, well, that's a technique used by human divers. It's not a very good one, but yes, of course. But the problem is the damage is already done, right? So if the bubbles, when in a beaked whale, for instance, they come up to the surface, they're racing up to the surface, and then you have this massive bubble formation, and they disrupt tissues, they go into the brain, and they explode small arteries, and the damage is done. Right, so you have internal hemorrhages everywhere, and the animal just dies. I mean, and they don't know physics, so they don't know, well, I must go down to decrease the size of the bubbles. But even if they did, the damage was already done. Did you understand, Laura?
Yeah, thank you.
Okay. Stephanie?
Yes. According to the lungs that are getting flat,
Yes.
the way to say, because in free diving, there are for some recent research on humans that show that because we thought we could not go, the human could not dive deeper than 30 meters or 100, I don't remember. Then they made research that they thought that after this point, like because the lungs get flat so much, it's not possible. But actually, what I hear is that there is a physiological thing going on, that the lungs get filled with blood. So all the vessels around the lungs get really filled with blood. So it increases the size basically of the lungs, so you don't have that problem. So because then for me the image of getting flat is a bit weird, and that's why also when this happens there is something changing in the homeostasis of the body, and that's why you need to pee usually when you go to the surface, because all your body's like the liquid stuff are a bit weird. Yeah. I don't know, I just wanted to add that. I don't know if you, yeah.
Yes, thank you. Well, yes, that is important. I mean, what happens is that the volume of air is extremely reduced. So in those persons that go really deep, what happens is that all the air is probably located in the nose and in the ear area. All the air, it's just not much, it's 5 liters. So it just collapses, but of course then in the lungs there's no air. But I've also heard that then you have all the plasma and lymph going into the how do you call it, alveoli, the little
alveoles.
Alveoli, the little sacs. Yes. And in addition, in marine mammals, what you have is big mucus. So the inside of these alveoli have mucus that stops the walls to glue. That's also one of the reasons why you have these massive clouds when the whales come up to the surface. Most of these mucus is just expelled when they're breathing.
Okay, thank you.